It is my great pleasure to open the Copernicus Lecture. And when opening it, I would like to say a few words about two people who are the most important people tonight, Nicolaus Copernicus and Joseph LeDoux. Well, Nicolaus Copernicus is quite all right to be with company with Joseph LeDoux. Of course, about Nicolaus Copernicus, we could say a lot of things and spend all the night commemorating his achievement, but I will focus only on one small episode, which I must say always struck me when I think about Copernicus and Kraków. As you know very well, Copernicus was student here at the Jagiellonian University in Kraków, and when he studied here in Kraków, he managed to buy two books, only two books. At that time, books were extremely expensive, and he managed to buy these books only because his rich uncle, who was a bishop on the northern part of Poland, gave him enough of money. At that time, one book was as expensive as approximately one village. And these two books, which he bought when here in Kraków, these two books are now preserved in Nuremberg in Germany, are the following two books. The first one is the Alphonsine Tables, and the second one is Euclid's Elements. Alphonsine Table is a sort of collection of astronomical data of that time, the most exact astronomical data of that time, position of stars. And Euclidean elements are, of course, the basis of geometric language in which the book of nature is written, as remarked by Galileo. And these two things, observational basis, experimental basis, and theory, are two legs on which every science, serious science, stands. Also the neuroscience. And here is where Joseph LeDoux enters the stage. He is, as we know, a neuroscientist and a professor of neuroscience and psychology at New York University. His work is about study of the neural basis of emotions, especially fear and anxiety. His work has elaborated in detail how the brain detects and responds to danger and learns and forms memories about the threats. Joe LeDoux has received a number of awards, including the Thyssen Foundation International Prize in Cognitive Science, the Jean-Louis Signoret Prize, and the Presidential Citation for contributions to the study of the emotional brain from the American Psychological Association, only to name a few. He is the author of several best-selling books, among others, The Emotional Brain and The Synaptic Self, How Our Brains Become Who We Are. Joseph LeDoux is a fellow of the American Association for the Advancement of Science, a fellow of the New York Academy of Science, and a fellow of the American Academy of Arts and Sciences. And last but not least, Joe LeDoux is also a member of the Amygdaloids, a band of scientists that plays original songs about mind and brain and mental disorders, the lyrics of which are often inspired by his research. And now we will listen to his talk about our emotional brains, and let's guess whether it will be in the form of a standard lecture or perhaps kind of lyric and music. I invite you.
Thank you, Michael. It's wonderful to be back in Krakow. It's been 13 years since I've been here. And thank you for organizing this conference. And Boraz, also thank you. And Jacek is the thread that ties all this together, because when I was here 13 years ago, I met him. He ended up coming to my lab to do research, and now he's grown on to become a resident in psychiatry at NYU. And I'm glad to be back here with him and with other friends from NYU and colleagues and friends from all over the world. It's a great pleasure to be here and to be talking to you this afternoon about a topic that I care a lot about, the emotional brain. And so let me just talk at the beginning about why we should care about emotions. I mean, for one thing, they make the significant events in our lives worth living. They mark these events. A life without emotion would be no life at all. In many ways, we are our emotions. And yet, in spite of this, emotions isn't always a topic that is popular in psychology and neuroscience. Why should we care about how the brain makes emotion? I mean, can't we learn everything we need to know about the mind without going into the brain? That was an approach that has been popular from time to time. But I think that we can understand a lot by studying things through the brain. For one thing, understanding the emotional brain enriches our understanding of who we are. I mean, if there's something we can learn about how our minds work by studying the brain, then we should go in that direction and try and figure it out. Pathological emotions, which are the cause of much human distress, are associated with alterations in the brain. And if we can figure out how these alterations are occurring, perhaps we can develop better therapies, both psychotherapies and pharmacological and perhaps even other therapies in the future. So let me just talk to you for a few minutes about how I got interested in emotions. I was a graduate student in the 1970s, and that was a time when there wasn't very much work on emotion, especially in neuroscience. There was work in psychology, but it wasn't really having much of an impact in the study of the brain. And like most people at the time, I thought that emotions were the center of mental life and behavior. So, that is, most lay people, but not most scientists. So I decided that that was something I wanted to get involved with, trying to figure out how the brain makes emotions. So why was there so little work on emotions at this time? I think there are a couple of reasons. One, in psychology and brain science had both sort of been converted by the religion of cognitive science, which didn't view emotions as a very interesting or important topic. I guess I probably shouldn't say it that way. That emotions were thought of as a topic that was outside the domain of cognitive science. So it just wasn't being studied under the idea of the computer metaphor and so forth that dominated cognitive science. But the other answer is that there was already an explanation of how the brain makes emotions, and that seemed to be perfectly good. This was the so-called limbic system theory, in which the brain was divided or the higher parts of the brain were divided into two parts. One was the new part of the brain, the neocortex, which is the part of the brain involved in our thinking, planning, decision-making, and so forth. It's where we have language and cognition. And then older parts of the cortex that are layered below the neocortex and presumed to be evolutionarily older. I don't have a pointer, but you can just, if you follow the seahorse there, which is the hippocampus. Hippocampus was based on the Greek word for sea monster, so the seahorse is there. If you follow it around, all the way around, it wraps around under the neocortex and forms a rim. And rim is the Latin word for rim is limbus. And so this became the limbic cortex, the old cortex or limbic cortex, because of this structural characteristic of it. And at some point, it was concluded that this might be the seat of our emotions. This conclusion was made by Paul MacLean. It was the culmination of a lot of ideas and research that had come before. And MacLean's idea was that this old cortex and subcortical areas that are connected with it, like the hypothalamus, which is just above the seahorse there, HYP, the amygdala, which is a little bit above there. It's not labeled very prominently. It wasn't a very big part of the limbic system. these subcortical structures connected with the old cortex formed what MacLean called the limbic system, which was the seat of emotion. He said the limbic system was involved in emotion and not cognition. The neocortex was involved in cognition and not emotion. Both of those things quickly became false, and yet the idea of the limbic system persisted. The limbic system was actually a pretty good hypothesis about the separate evolution of certain higher cognitive functions like language and thought as distinct from emotion, which predates these higher cognitive functions evolutionarily. But it ran into trouble. For one thing, it was never unequivocally defined as to how you would identify what a limbic area was and what it wasn't. So pretty soon, the original definition of areas connected with the old cortex began to run into trouble when it was discovered that parts of the so-called new cortex exist in pre-mammalian form. So they're precursors to what became new cortex in reptiles and birds, which sort of throws a monkey wrench into the whole thing, because new cortex was supposed to be new with mammals. And there are a variety of other problems. One was many limbic areas were defined on the basis of connectivity with the hypothalamus. But once it was discovered that so-called neocortical areas are connected with the hypothalamus, as well as the lowest parts of the brain and even the spinal cord, the limbic system, by that definition, began to include the whole brain. So in the absence of a clear way of defining what the limbic system is, it's been hard to really accept that as a firm conclusion. Another problem is that it didn't really tell us anything about how any particular emotion was mediated by this system. It simply said that emotion in general was mediated by the limbic system. So it tried to explain all emotions at once in one kind of big idea. And while that's a laudable goal, it really hasn't worked very well, because as you look through the limbic system for an explanation of fear or joy or any other emotion, you can't really find it. So let me now just backtrack I want to go through a couple of themes that were apparent to me when I first got started doing this work, and that I still sort of adhere to. One is that I decided, because the limbic system had failed to explain emotions by being a theory of all emotions, that I was going to work on one emotion, and the emotion I chose to work on was fear. That way, I didn't have the problem of defining emotion, which had never really been defined in the limbic system theory or other brain theories very well. So instead of trying to define what emotion was, I just picked one thing that everybody agrees is an emotion, which is fear, and decided to study how the brain does that. And one good reason for doing that is that there are very good tasks that you can use to study so-called fear responses in an animal brain. And this allows us to go into the brain and understand a lot about how the brain is making something like fear. And the other theme was that I had the idea of borrowing the information processing approach from cognitive science to try and think about emotions like fear not as a subjective state of experience, in other words, not the feeling of being afraid, but instead as a way of studying how the brain detects danger and responds to danger. So there must be some kind of emotional processing network that detects the danger in the world and then orchestrates responses that produce those responses. So this is totally compatible with the information processing approach in psychology, but we're applying it here to emotion rather than to cognition in general. So, this raises the question of what is emotion. James Russell is one of the participants in this conference, once wrote that everyone knows what emotion is until they're asked to define it. And that's kind of the way things are. The other famous comment that somebody may know who said this, I can't remember at the moment, but there are as many theories of emotion as there are emotion theorists, which kind of states the state of we'll call it art at this point rather than science. But yet there are some things that, that we, if we go back to this idea that everyone knows what emotion is until they are asked to define it, let's just say everyone knows what emotion is. So you probably can guess what this emotion is. I'll just assume you know it's sadness, and you can see that in this person's face. You can see it in a baby's face. You can see it in a mask. It doesn't have to be a real human. You can see it in line drawing. And you can even see it in body posture. So it's not just a facial thing. It's things that, it's something that you can identify in a variety of ways. But what exactly are we talking about with sadness? So we have this inner feeling that each of us can individually experience, and then we have an outer response that we can measure. Now, long ago, William James described these, popularized this distinction. It probably existed long before, but this distinction between emotional experience or feelings on the one hand, and the expression of emotional responses, bodily responses that happen at the same time that we have these experiences, like a behavioral response, these expressions of emotion, and accompanying changes in the autonomic nervous system and stress hormones and other things that may be released at the same time. So this raises the question of whether there is a core thing called emotion that's accounting for both of these things. So we use the term emotion for both of these bodily feelings and for the bodily responses and for the feelings. But as far as I can tell, there's no one kind of core thing that really connects these in some kind of obligatory way. For one thing, we can, in studies of humans, you can show that people can have emotional responses to stimuli that they don't see or experience in any way, and without ever having any kind of feeling about it. So, activation of the systems that produce emotional responses don't cause necessarily bodily responses. And we can also have internal feelings that don't get measured or don't get expressed in any way that we can obviously detect in some cases. So what I want to do is just sort of maybe separate these things out. Also, the correlation between bodily responses and feelings, people have measured these things like Peter Lang and others, and he finds that these correlations are very weak. So let's throw out the hypothesis that the brain mechanisms that underlie feelings and bodily responses are distinct. They can interact directly, but they're separate things, and we need to separately think about them. So if this is true, there's no emotion system. There are systems that are responsible for various things that we label as emotional, but there isn't an emotional system per se. So let's start with feelings. So understanding feelings is a very important thing. Obviously, it's, as humans, this is what we think of emotions as in many ways. Animal studies are essential for making progress in terms of detailed brain mechanisms, but we don't have any way of really studying feelings in animals. We don't even know if animals experience anything. I mean, you can probably assume that they must have some kind of experience, but as the philosophers have told us, for example, Thomas Nagel in his famous book, What Is It Like to Be a Bat? Well, it's like being a bat, something that a human can never understand or experience, because only a human can only know what it's like to be a human. A bat can only know what it's like to be a bat. So if a bat has feelings or a rat has feelings, that's something that is peculiar to that organism and can't be penetrated by the mind of another organism. But even if we could be sure that animals had feelings, study of animals would only tell us about human feelings if the mechanisms that make human feelings possible are also present in the animal brain, and if they're the same mechanisms that make feelings possible in the human brain. So, but from what we know about the mechanisms of consciousness in general, this may not be the case, because areas of the brain, like the dorsolateral prefrontal cortex, that tend to be, for example, if you do imaging studies of people and show them stimuli that they don't recognize, or they aren't consciously aware of, the prefrontal cortex isn't activated. But if they're allowed to be aware of the stimulus, the prefrontal cortex is activated. This is true of emotional stimuli or non-emotional stimuli. So because this dorsolateral prefrontal cortex is especially well developed in humans, less developed in other primates, and believed to not be existent in other mammals, we have reasons to be suspicious about the nature of feelings in other animals, that if they do have feelings, they're not likely to be the kinds of conscious experiences that we have when we have a feeling. So feelings have to be studied, I think, in people. This is something that we can and should do in people, and something that may not be the most profitable thing to pursue in animal studies. What about emotional responses? Well, behavioral and physical physiological responses can be studied similarly in humans and animals. So this is something that we can definitely profitably study in animals and really make some progress and be able to equate things across animals and humans. So although these are not necessarily totally reliable or valid indicators of human feelings, they are useful in indexing the activity of the brain circuits that control them. The brain circuits that control these emotional responses, what I'd like to do is just call these survival circuits for the moment. So survival circuits include at a minimum systems involved in defense, energy, nutrition, maintenance, fluid balance, thermoregulation, procreation. The goal of each of these systems or circuits is to evaluate or appraise the significance of a current or an anticipated challenge and to trigger responses that are appropriate to the computed meaning of that stimulus or situation. So each circuit is tuned to certain kinds of classes of environmental and or internal physiological stimuli. In the presence of such stimuli, a set of more or less innate responses and physiological adjustments are released in the effort to cope with this immediately present challenge that the organism is facing. These stimulus-response connections are implemented by computational networks that evaluate the stimulus value on the basis of innate programming. So there are certain things that will elicit these kinds of evaluations innately in people. You run across a snake on a path, it will probably startle you and scare you a bit. A baby's crying, other things that are, we don't have a lot of innate programming in humans, but we have some things that are innately programmed to elicit emotions in us. So on the basis of these innate program, or on the basis of past experience that leads to the formation of associations between innately significant events and novel stimuli, these survival circuits are activated. So these computations are circuit specific, so that if you activate a defense system, a nutritional system, or a procreational system, different systems will be activated by different kinds of stimuli. The triggers of fear don't activate don't activate the system of eating or sex. These survival circuits are highly conserved in mammals and constitute a kind of species-independent way of defining emotion systems. Now, you may be saying, well, those things you talked about aren't emotion systems, but I'll address that later. So here's an example of a very highly conserved emotion circuit, or survival circuit in the mammalian brain. This is the sexual or procreational circuit in mammals. The hypothalamic and other forebrain areas involved in sexual behavior, the detection of sexual stimuli, the elicitation of responses on the basis of these stimuli, all these computations are the same throughout mammals: horses, dogs, cats, rats. In spite of this, the responses aren't necessarily always expressed the same way. So in rats, the female has to adopt a posture called lordosis, where she sways her back in a certain way in order for the male to penetrate. If she doesn't do this, she can't, the male can't penetrate her, and they can't have intercourse, and she can't be impregnated. So in order for her to do this, she has to be in a certain hormonal state, which only occurs during her cycle. If she's not in that part of her cycle, she can't have sex. This is not the case in primates. Primates can engage in sex anytime, but they have relatively stereotyped behavioral postures as well, somewhat different than what you see in rats. Each species basically has a somewhat stereotyped way of responding, engaging in sex and feeding and so forth. So rats do it one way, monkeys do it a different way, people do it all sorts of ways. And so the point is that the way the responses are expressed is not what's conserved across species, but the circuit that is controlling these activities is what's conserved. That's why, in a way, the circuit is perhaps the only element that can tell us about the conserved nature of these systems across evolution. We can use the behavior to access these systems in a species-specific way and in a species-general way, but we have to recognize that across species the responses may differ, even though the circuits are highly conserved. Now, that's not to say there are no subtle differences in the brain that don't differ, because obviously for the female rat to adopt the lordosis posture, there has to be something that makes that happen. But in general, the circuits, I believe, are more conserved than the behavioral responses themselves. So I've already said that. So what I want to argue is that neither responses nor feelings are a good way to characterize emotions in a species general way. Now, this is just a sidetrack. This is work from Lin and David Anderson. This is showing two of these survival circuits in action. What they're able to do is to use special metabolic marking techniques. In this case, they're activating gene expression in response to either, they're having mice either fight or mate, and they'll either mate first. So this is a male, so he's either put with a female and he mates, or he's put with an intruder and he fights, or you do it in the other way, and it's all counterbalanced and everything. But this is looking at the role of the hypothalamic areas in certain hypothalamic areas in mating and fighting. It's kind of like marriage. You mate, you fight, or you might fight then mate, or whatever. So it's the interaction of these two systems, and they're able to go at it in a very precise way. So what you can see is that where the arrows are, we've got mating on the bottom row and fighting just above it, and then the control group is on top. So look in the second column from the right as you're facing here. You see that the ventromedial hypothalamus there is activated in both cases. So you might be tempted to conclude that this area is involved in both mating and fighting. But using very precise techniques, what they were able to do, and I won't go through the details of this slide, is they're able to show that different cells are involved in mating and fighting. So even though the same area is activated, it's different cells that are involved in mating and fighting. So why this is relevant is that if this were, for example, a functional imaging study, you would see a blob there, and you would see the same area activated in both cases, but underlying all that would be some very specific cellular responses, and each of those cells may have different kinds of synaptic contacts. This is not a put down of imaging at all. It's just we all recognize the limitations of imaging. But what it suggests is that we really have to do this kind of animal research to go into the details of how specific circuits are operating, because even within an area, there are cellular differences and molecular differences perhaps that account for the different behaviors that this particular area is involved with. So, for example, if we see something like the amygdala being activated in fear and joy, rather than concluding that the amygdala is a universal emotional computer, what we should do is go into the brain and study whether the joy circuits and the fear circuits are actually the same within those blobs. And there's a lot of work going on now that is attempting to do this kind of thing. I focused on the fear angle, but other people are working on the positive or appetitive angle. But what I want to do now is move away from these words, because I think the words are going to get into the way as we begin to try and study these emotion words, as we begin to try and study these things in more details. So, but let me go through a few more things here first. These survival functions have their origin in primordial single-cell organisms in some cases. For example, here we have a petri dish. It has bacteria in it. And you probably can't see this very well, but in the top panel, the bacteria are sort of randomly dispersed. But if you squirt acid, oh, yeah, thank you. It doesn't really, it does, okay. Anyway, on the top, the bacteria are dispersed. On the bottom, someone has squirted acid in there, and they all move to the side. So obviously, the bacteria aren't afraid of the acid. Their nature is such that they have the ability to detect harmful chemicals in their environment, and also to detect useful chemicals, because they have to have nutrition and energy and all of that to stay alive as well. So these kinds of basic functions of survival go back to the earliest forms of life. And as you evolve more complex forms of life with specialized cells and specialized systems, it's only an elaboration of this primordial life-sustaining capacity. In people, obviously, you pour acid in the swimming pool and they all move out as well. Do they move out because they're afraid, or because they're like the bacteria, which they have these detection systems? Obviously, this is kind of a trivial example, and I don't mean to make too much about it, except to say that the brain can detect danger and respond to danger without the mind having to be engaged. So again, as I said, survival circuits are highly conserved in mammals. In fact, throughout vertebrates as well, there's a lot of this conservation. And here's some examples. This is kind of contradicting what I said before about the responses not being conserved, but it's not that responses aren't conserved, but they're not always conserved. But here's some examples of, I don't know what the shark is doing here, but it looks like the same thing anyway. So, okay, one more. All right, so anyway, this animal is obviously expressing displeasure or unhappiness or rage or whatever in terms of whatever else is out there. And so is this guy, and so is this guy. So we have these things are really hardwired into the nervous system. And as I said before, the stimuli that elicit these responses can be either innately programmed or can be learned from experience. And I've already said this about the outward responses not being universal. But this is one kind of response that is fairly universal throughout mammals, the fight-flight response involving, it's controlled by the brain. Obviously, the brain detects danger. Hormones are released. The autonomic nervous system is activated. Muscles are tense and so forth. All of these responses occur in a very stereotyped way because there's a system in the brain that's orchestrating all of this. So what are some of the consequences of activating one of these survival circuits? Well, I just told you some of the consequences. One, these downstream areas involved in the control of behavioral, autonomic, and endocrine responses in the periphery are activated. Another is that arousal systems in the brain, such as monoamine systems like norepinephrine and dopamine, serotonin in the brainstem are activated. These release their chemicals throughout the brain and cause a state of arousal or activation within the brain. You also get the activation of motivational systems involved in goal-directed instrumental behaviors. And Nico's talk this morning at the conference made me think about something else that I need to build in here, which are these more impulsive behaviors as well, which are goal-oriented but not pre-programmed or in advance in a way. Now, the motivational systems are interesting because we don't usually think of activation of motivational systems as emotional, but motivation and emotion are continuous. An emotional stimulus elicits this brain activity and bodily responses, and then your brain finds itself in the state of danger, and you're now motivated to take action, to do something about it. You're going to run up the tree, you're going to run away, or you're going to do whatever you have to do to deal with the danger. So emotions elicit motivations, but being in a motivational state can also elicit emotions. The activity of all of this is monitored and assessed in cortical areas involved in working memory, like the dorsolateral prefrontal cortex and other areas, and integrated with information retrieved into working memory about this current state, the external context, long-term memories, and so forth. And all of this is put into a mental conglomeration that is, I think, what a feeling actually is. It's the representation of all this stuff about what's happening in the brain, in the body, in the environment, the memories being retrieved, and all of this put together, labeled as a fear or whatever, and that's what our emotional experiences may be, or at least could be a large part of it. This widespread activation and coordination and monopolization of brain resources occurs. So in a situation of danger, your brain needs to put all its eggs in the basket of staying alive. So everything gets coordinated, many brain systems are active, are working together to ensure that you survive this moment. The body's systems are co-opted to help deal with this danger. So you have blood in the fight-flight response, you have blood moving from the gut and skin, areas that don't need the blood at the moment, to areas that do need the blood, like the arms and legs to perform the fight or flight response, for example. And so through this kind of widespread coordination and integration, the survival of the organism is enhanced. So while the survival circuit activity is correlated with feelings in humans, feelings are the result of this separate evolutionary force that's made it possible for the brain to be aware of its own activities, its own behavioral responses, the environmental context in which all of this occurred. But both fall under this rubric of emotion, we end up with a lot of confusion. So I think by separating the mechanisms of feeling from the separation of the survival, from the survival circuit mechanisms, the study of motion may move forward in a more coherent way. So one thing I think that's important is, there was a time in neuroscience and psychology where these homeostatic mechanisms had a prominent role, but they've sort of like lost favor as being just sort of physiological underpinnings, not very interesting, not where the action is. But I think it's important that we, maybe bring homeostasis back to the forefront. Homeostasis also has lost some value because of the concept of allostasis, which is the change in homeostatic values as stress develops and so forth. But I just want to make a plea for the return of homeostasis in a way. So these homeostatic mechanisms are carried out implicitly or unconsciously. If survival circuits are an important component of our species' general aspects of emotion, then we're never non-emotional. Just to address Yadin's comment this morning in his lecture. He asked the question, are we ever non-emotional? I would say, perhaps not, because these systems are always monitoring the environment, monitoring our internal states, adjusting things. So we're constantly making adjustments, even though we're not consciously aware of those adjustments. So if emotion is used to refer to conscious feeling states, we're never non-emotional during these homeostatic processes, but we can become emotional when the brain becomes aware of survival circuit activity through either internal processing in the brain, feedback from the body, observation of one's own behavior and the physical and social context of the moment, the memory of the context, the memory of past context and stimuli, and the predicted context of the future. So all of this gets put together, and it's this monitoring or this awareness of all these things that are going on that constitutes the feeling. And this is, I think, something that is, as I've said several times already, this is very different from the evolutionary system, the systems that evolved to generate these responses that ultimately participate in feelings. But a feeling is much, much more complicated and involves many other processes. So let me give you some objections, and I probably don't have them all, but here are a few objections to some of the things I've talked about. One, it's, you might say, well, I've just repackaged basic emotions theory. So basic emotions theory tries to find brain mechanisms that account for emotions, but it does this on the basis of words that are derived from our introspective experience. So we look for a system in the brain that's responsible for fear, joy, anger, love, happiness, sadness, surprise, disgust, and many other things. What we often do is, if you're a basic emotions theorist, you would use a person's ability to characterize these emotions on the faces of people to identify, which emotions can be recognized around the world. Now, as James Russell has pointed out, many of these kinds of studies are done by giving people a multiple choice of, is this anger, sadness, or fear? And if instead of giving them a multiple choice like that, they're just asked to spontaneously generate the emotion or identify what the emotion is, their performance drops way down. So, people can identify these things, and if the examples are really, really stereotyped, I think it's probably fairly easy. But there are some problems with this approach. And the main problem, though, is that we're using these words derived from our introspective experience to go looking in the brain for things that began to evolve with single-cell organisms. And I think this is why should we expect the most complex function of the human brain, language, and the newest part that's based on the newest parts of the brain in evolution, to be able to access these evolutionarily old systems that have been around much, much longer. So I've already said this. So as bad as this might be, if we're looking for emotion systems in the human brain, it's totally useless if we're going into the rat brain. How do we find joy or love or happiness in the rat brain? We can't. But what we can do in both a human brain and a rat brain is study these the survival circuit functions: defense, energy, nutrition, fluid balance, thermoregulation, procreation. And I'll tell you why that's important in a minute. So standard emotions, another objection might be that standard emotions are not included in my list. No anger, no joy, pleasure circuit. As you see here, there's no anger or joy, pleasure circuit. But aggression is, in the view that I'm trying to promote here, is defined by the context in which the aggression occurs. So aggression in an attempt to protect oneself from harm is a function of the defense circuitry. Aggression related to competition for mates, part of the reproductive circuitry. Aggression towards prey species, part of the nutrition circuitry. So aggression is not one thing. There are lots of different kinds of aggression, as we heard in the last lecture in the conference earlier today. So why would there be an aggression circuit? There isn't an aggression circuit. There are aggression circuits related to specific forms of aggression. Same with joy and pleasure. There's no joy-pleasure circuit. There are aspects of joy that are involved, that are related to eating, drinking, sex, and so forth. And these are reinforcing events. But they're reinforcing events related to specific homeostatic mechanisms. And that's where they derive their reinforcing value from. So what I'm trying to do is reclassify things on the basis of these survival circuits, basically. So by focusing on the subjective state, anger or joy or pleasure, other theories are glossing over these underlying details of emotional processing for the sake of converging on a single word that symbolizes diverse underlying states mediated by the different circuits. Okay, other objections. Complex emotions aren't included. I don't have empathy, jealousy, remorse. But unless these can be shown to be based on circuits that are conserved throughout mammalian species, they're not part of the account that I'm trying to deal with here. If they can, then we can account for them in some way. What about emotion and motivation? I've already talked about this, but I mix them together. So eating and drinking circuits are usually discussed in the context of motivation, not emotion. But motivation and emotion are always intertwined. Emotional stimuli are motivating. Motivational states alter the sensitivity to emotional stimuli. They're called incentives when this happens. And the relevant motivational state, the stimuli that related to food, grab attention. When we're hungry, we ignore these stimuli. When we are hungry, we attend to these stimuli, but when we're full, we ignore these stimuli. So the activation of a motivational system will tune your antenna to certain kinds of incentives and make you more aware of those. These incentives are basically emotional stimuli that are going to be processed by these emotional and survival circuits and activate these hardwired responses that then become consumatory responses. The incentive part is the part where you're looking and trying to satisfy the motivational state that's arisen, and then you consume it by these hardwired responses. All right, let me just summarize some of what I've gone through so far. So brain circuits involved in key survival functions are highly conserved in mammals. Included are defense, nutrition, fluid balance, procreation, thermoregulation, etc. These circuits and their functions are organized similarly in spite of the fact that they can control different behavioral responses in different species. When one of these circuits is activated, a global synchronization or monopolization of brain resources occurs. The circuits evolved not to create feelings, but to perform these survival functions. The global activity triggered by the activation of these circuits is what emotion refers to. Motivation and emotion are continuous. I won't repeat that since I already did it. Feelings result when consciousness witnesses one of these global states. The nature of feelings in an organism is a function of the kind of conscious capacity its brain has, with the possibilities ranging from none in a single-cell organism to a lot in humans. Feelings in humans are greatly influenced by our capacity for semantic categorization provided by language. The particular circuit that's active can be instantaneously overridden by activity in other circuits. So defense normally trumps feeding, but if deprivation is severe, then feeding overrides defense. So if you're really hungry, you have to go out into the world and take a chance that you might be eaten by somebody else to get food. So circuits can also be partially inhibited and can also be coactive. As Nico mentioned this morning, you can have multiple emotions or multiple motivational states simultaneously, which leads to complex multiple emotions. So, all right, so that's most of what I wanted to say that is relatively new. Now what I want to do is talk about one particular example of one of these survival circuits, and that's the defense system. Now the defense system its job is to detect danger and control hardwired responses that help the organism stay alive. So I'm switching gears now and not going to be talking about, even though most of my scientific career I've talked about fear, what I want to do is talk about defense rather than fear, especially in the context of what I've just been telling you. What I want to do is try to avoid implying that we're studying these fearful feelings in animals or people. And much has been learned about this defense system using Pavlovian fear conditioning, in which a neutral stimulus acquires the capacity to elicit defense responses after it occurs with an aversive stimulus. Pavlov initially called this defense conditioning, but later researchers turned to fear conditioning as the terminology. So we're going back to Pavlov's original terminology here. So here's defense conditioning in action. So the rat's in a chamber. It hears a sound. Nothing much happens. If the sound is paired with an electric shock, then after a few pairings, one is sufficient, then the rat will express these fear responses, defense responses to the sound itself. So some of the characteristic responses are freezing behavior, autonomic nervous system activity, the release of stress hormones. These occur after conditioning, but not before, to the tone, and they occur in the presence of a cat or in the presence of a tone after conditioning. So the responses, a rat or a person doesn't have to learn how to be afraid or doesn't have to learn how to defend. What it has to learn is what to express defense in the presence of it. We learn what stimuli are dangerous. And that's the key thing that we want to study through this defense conditioning is what stimuli or how stimuli acquire these aversive meanings. So this kind of defense conditioning is universal throughout the animal kingdom. I think every animal that, I don't know of any animal that's ever been studied that hasn't shown this kind of conditioning. The first example that I know of in the fly was by Yadin Dudai, one of our distinguished participants here. Yadin studied this in terms of genetic alterations in fly behavior. Eric Kandel's Nobel Prize-winning work was based on the marine snail. All the rest of these are vertebrate animals. And in every vertebrate animal, including humans, and we'll hear more about the human brain and defense conditioning from Liz Phelps tomorrow. But every animal, oops, this is, all right, that should say brain. Somehow the translation didn't occur there. We can follow the way this works by tracing the conditioned stimulus through the brain to the conditioned response. And in every animal that has been studied that has an amygdala, the amygdala is the centerpiece of the system. The conditioned stimulus is processed by the amygdala to produce the conditioned response. So this is the appraisal system in the brain here for this kind of emotional stimulus. So the stimulus is detected, the responses occur. It doesn't matter what kind of stimulus you use, and we heard about Regina's studies of odor conditioning earlier, but tones, lights, context, odors, all can be used. It doesn't matter what you measure as the output response of the behavior, autonomic, hormonal responses. It doesn't matter what kind of animal you do the studies in, the amygdala is a key part of this defense conditioning system. Now, these guys don't have an amygdala, so they learn, they do their defense conditioning in a different way. So, we could put the chain of events even further back. I don't know if a bacteria learns anything, but somewhere between single-cell organisms and other invertebrates, this ability to detect danger and learn about novel dangers by forming associations between dangers and novel stimuli appears in early invertebrate evolution and somehow is continued in vertebrate evolution through the presence of an amygdala in fish all the way through humans. So the amygdala is here in this example. So the hiker's walking through the woods. He's about to step on the snake. He freezes. His blood pressure and heart rate begin to accelerate. The stimulus goes into a way station in the lower part of the brain called the thalamus. and then it will go to the cortex where the person can perceive the snake, but it also gets to the amygdala directly from the thalamus. So this provides a quick and dirty input to the amygdala. In this case, we see something that's roughly snake-like, but it could be a stick on the ground rather than a snake. And the person in this context would probably also halt if you saw, if you were walking along and there was a curved snake or a curved stick on the ground, you might halt over that. So the idea is that through this pathway, we can't distinguish a stick from a snake, but evolutionarily, we're better off treating sticks as snakes than snakes as sticks, so we don't step on it. And then through this pathway, we can evaluate whether it is, in fact, a snake. Now, sometimes we talk about these as unconscious and conscious pathways. My thinking on this has evolved considerably since I published this in The Emotional Brain in 1996. I think probably all of this is unconscious, and until the information reaches prefrontal cortex or other neocortical areas, it isn't represented in consciousness. But in order to get to the prefrontal cortex, it has to go through these visual cortex areas, not to the amygdala, but from these visual cortex areas to prefrontal areas. So while the cortical areas are the route to conscious awareness of the stimulus, the cortical areas are not necessarily, the cortical processing of the snake is not necessarily a conscious stimulus that activates the amygdala. All right, so the amygdala, is that really a real thing? The amygdala has a lot of different parts. There are at least a dozen different nuclei within the amygdala, and this is just a few of them shown here. And two of them that are particularly important are the lateral nucleus, here, the LA. It's a triangular structure sitting here at the top of the amygdala in the rat, and then the central amygdala, which is a circular structure here. Now, we can study these things very well in the rat brain, but the techniques for studying these in the human brain are not quite there yet. Paul Whalen and Liz and others are attempting to be able to image these different nuclei in the human brain, but we aren't quite there at this point. So in order to get to the nuclei, and certainly to get inside the nuclei into the cells and synapses, we still have to do animal research. But as you see here, damage to the amygdala, the entire amygdala, or the central or the lateral nucleus disrupts fear conditioning, but none of the other amygdala lesions has any effect. So really, we only need two parts of the amygdala, the lateral and the central nucleus. Sometimes we divide the amygdala into two parts, a basolateral and a centromedial, but these are not very useful characterizations. It's dividing the amygdala in half is not very, doesn't provide enough information because it's really the lateral and central that are key, not the rest of these structures. So information comes in from the outside world into the lateral nucleus. The lateral communicates with the central nucleus directly and by way of other intramygdala connections, and the central nucleus controls the outputs. So damage to the central amygdala disrupts all of these fear responses. Damage to areas that the central amygdala is connected to disrupts one or the other response, but not all of them. But things get even more complicated because it's not the entire lateral amygdala that's involved in the formation of the association. So in order for learning to occur, this is true of any kind of associative learning, there has to be some way in the brain for the two things that you want to associate to come together. So in the case of a tone and a shock, or a CS and a US, the two stimuli have to meet up somewhere. In one place they meet up is in the lateral amygdala, but only in the dorsal half of the lateral amygdala. So the CS and US converge in the dorsal half, but not in the ventral half. So we started with the brain. We identified the amygdala as key. We identified the lateral amygdala as important because it gets the sensory input. And now we've divided the lateral amygdala into two parts. And it gets even worse because within the dorsal half of the lateral amygdala, here's the dorsal half of the lateral amygdala again, we have two populations of cells. One that we call trigger cells, that's located in the apex here of the triangle. These cells learn rapidly and then reset. They go back to baseline. Then right below them are these storage cells. These learn more slowly,
but they hold on to the memory. See, these never go back to baseline. They stay elevated. These are back to baseline. But these stay elevated. And what's important is they stay elevated after the rat has been to therapy. So the rat has been given cognitive behavioral therapy or extinction training, and here he no longer is expressing fear responses. The therapy was successful. So he's not showing any fear to the, or I shouldn't say fear, I have to say defense now. He's not showing any defensive behavior here, but his amygdala cells, these lateral amygdala cells down here in the ventral part of the dorsal half, still remember the danger. This is important because people with phobia can be successfully treated, but then the phobia can always be brought back by stress, and this is true in rats as well. So phobias are always in the brain once they're acquired. It's just a matter of whether they're accessible or not by the outside world. So again, the trigger cells and the storage cells. Now, to learn more about how all this works, we can take the amygdala out of the brain, put it into a dish, and study it artificially. So we can create learning in the amygdala by making this amygdala cell, this black dot here, think that it's getting a tone and a shock. So as far as the cell knows, the rat is hearing a tone right now. All we're doing is electrically stimulating the pathway that brings the tone to the lateral amygdala cell. So obviously a cell doesn't know whether it's in a dish or in the rat in the real world, and so it's just getting synaptic inputs in either case. So we're synaptically activating the cell while at the same time it's getting depolarization by injecting current into it. So when the rat gets a foot shock, these cells, these amygdala cells, will fire a strong response. In other words, an action potential is generated in the cell because the calcium comes into the cell, and sodium comes in and triggers the action potential inside the cell, causing a strong electrical response. During that action potential, the influx of calcium allows a weak input to strengthen. So this is what's often called the Hebbian hypothesis, although the great Polish neuroscientist Konorski also had a similar idea around the same time. So according to the Hebbian hypothesis, weak inputs that don't normally produce these action potentials become strengthened if the weak input is activating the cell at the same time that the strong input is depolarizing the cell. So, sorry, I'm out of sequence here. Let's see if I can get to the right spot. Okay. So here we have a weak stimulus coming in. It's releasing neurotransmitter. The strong stimulus is coming in and activating action potentials. This is opening up channels that allow calcium to come into the synapse here, where the tone is. This is the tone synapse. This is the shock input. Calcium comes in. This allows the activation of certain enzymes that go to the cell nucleus, lead to gene expression and protein synthesis. The proteins come back and insert new AMPA receptors and do all sorts of things at the synapse that make this connection now stronger. So the idea is that the weak becomes strong by being associated with the strong. So the strong stimulus produces certain biochemical changes inside the cell that strengthen the connection that the weak cell normally makes. And through this kind of strengthening, an association is then formed so that the weak stimulus, the tone, when it gets to the cell, can now trigger an action potential in this cell and then initiate the activate the lateral amygdala, which can then, the information can then flow to the central nucleus, activate all of these responses, and the animal would then express the fear response. So this circuitry that we've identified here in the rat has been used to then go and look into the reptile brain to find whether a similar kind of circuitry is available. It's much harder to study these things in reptiles. The behavioral tasks are not as good. The anatomy is not quite as well worked out. So my colleagues in Spain took the anatomy that we've worked out in the rat and used, for example, connectivity with brainstem and other areas to identify what the central nucleus might be, and to use connectivity with sensory structures to identify what the lateral nucleus might be. This is not LA in the usual sense. This is the PDVR is the lateral amygdala of the reptile. The SAT is the central amygdala. And in spite of what we see here is a conserved circuit, the behavioral response that the circuit activates is different, going back to a point I made earlier. The survival circuit is the same, but in the lizard, what you see is something called tonic immobility or playing dead, which is more of a kind of limp body rather than a freezing response, which is a very stiff body response. So the same circuit can control different kinds of outputs in different species, but the circuit remains the same or is conserved even across reptiles and mammals in this case. All right, so can we, I want to talk some more about creating artificial memories, because this is an exciting new area. So we've been taking advantage of a new technique involved what's called optogenetics that Carl Deiseroth at Stanford has developed and popularized through various studies that he's been doing. And Josh Johansson in my lab began to use these techniques. And basically what you do is you take a light-sensitive molecule, in this case channel rhodopsin, and you couple it to a virus and inject that into the amygdala. So the virus then takes that into the amygdala cells, and then you take the cannula out, let the animal recover, let the virus get incorporated for a few days, and then you put another pipette into the amygdala. In this case, it's a fiber optic cable, a very tiny fiber optic cable. And when you shine the light in the amygdala, what will happen is the cells will now fire action potentials. So here's an example. At this point, each of these is a laser light impulse shined into the amygdala after the channel rhodopsin has been incorporated. And you see these very precisely timed action potentials. These are different stimulations. Each row is a repetition of this long stimulus consisting of multiple pulses. But you see that each pulse elicits action potentials. And if you then measure gene expression, in this case CFOS activation in the lateral amygdala, what you see is that on the side where the laser light was activated, the cells are showing functional activity, whereas on the other side there's no activity. So this is just a proof that we can do this kind of thing. Skip that. But the key thing is, can we now use this to create learning? I told you a few minutes ago that under this kind of Hebbian-Konarsky hypothesis, if you depolarize neurons while they're getting a weak input, then something happens that makes the weak input strong. So what we're going to do is get rid of the electric shock to the feet in the rat and instead just artificially depolarize these amygdala cells with the light while the rat's listening to a tone. This is a real rat, not a brain slicer. So the rat is hearing a tone and the cells are being depolarized by the light. And what you can see is that when this happens over so I can find the button again. Yeah. Over these 4 blocks of trials here, the rat learns to express these defense responses in the presence of the tone. So the first block, the CS is alone. By the second, there are 4 trials in each block. By the second block, the CS has been paired with the light several times, and you begin to see a freezing response. By the third block, you have more, and by the fourth block, you have some learning here that's pretty substantial. The control animals have the channel rhodopsin inserted, but the sorry, has the viral thing inserted, but it's just a fluorescent protein. No channel rhodopsin is inserted in there, and you see no learning. If you insert the channel rhodopsin and shine the light, but you don't pair it with the tone, you just separate those in time, you still give the same number of tones and the same number of light stimulations, no learning occurs there either. So we can create learning in this artificial way. Now, oops, I can go back. The learning is measurable, it's significant, and so forth, but it's not as robust as normal learning. So one hypothesis is that, this is based on the work of Kandel and others, is that in addition to needing this kind of Hebbian plasticity, you also need a neuromodulator to boost Hebbian plasticity to get normal levels of learning. So in other words, in a real brain, in a very aversive situation like this, in addition to the tone and the shock meeting up in the amygdala, the shock is going to activate brainstem monoamine systems like norepinephrine and so forth, and cause them to release their chemicals throughout the brain. These chemicals will then be released in the amygdala, where they can potentiate the Hebbian plasticity that's occurring. And so the question is, if we boost the monoamines during this kind of artificial stimulation, do we boost the learning? And let's skip that because I don't need to talk about that. If we do that, well, this is what this is showing is if we block the monoamines, we reduce the learning. And let's see if I have the other slide. No, I don't. Yes, and if we give a drug that enhances norepinephrine, we boost the learning. So we can either block or reduce learning or boost learning by manipulating the norepinephrine system simultaneously with this kind of Hebbian artificial learning process. So let me turn to another relatively new area of research. This is a study that Linnea Ostroff has recently published. And what you're looking at here is a pretty picture, but it's not just a picture. This is real data. So what she's done is used an electron microscope to take thousands and thousands of pictures in sequence of a neuron. Imagine this picture has lines going across it. So each line would be one picture. And so there are thousands and thousands of pictures going across here. And by using these electron microscopic pictures, stacking them up together, she's recreating a dendrite coming in at this synapse right here. Sorry, this is a presynaptic axon terminal coming in, forming a synapse on a dendrite right here. All these little blue-purple spots are neurotransmitter vesicles. So they contain neurotransmitter that will be probably glutamate that will be released when this presynaptic neuron is depolarized. The action potential invading the axon will release the vesicles into the synapse. The vesicles will open up, spill their transmitter into the synapse, bind to postsynaptic receptors here on the dendrite, and produce postsynaptic responses here. So we see a lot of things here. This is the spine apparatus. This is dendritic spine apparatus in the spine. The yellow is the smooth endoplasmic reticulum. The blue is the mitochondria here in this case. The color coding is not right. That's why it looks a little funny here. But while this is a nice picture, what she is showing in this study is that she can actually manipulate the structure of these neurons by doing this kind of defense conditioning. So in one case, what she's doing is increasing the size of the synapse by producing this fear conditioning or defense conditioning procedure. I'm sorry, this is the wrong side. So what we're seeing here is an increase in polyribosomes. These are protein synthesizing elements in the dendrite. So this suggests that there can be some protein synthesis in the dendrite, but that's not the main thing I want to say here. And this is neat. Let's go to this slide. Okay. So this is the key point, that these are small, but they're significant events. And what we're talking about here are small changes in the size of a spine. Now, a spine itself is a very small thing, but we're looking at not just the spine, but the postsynaptic density on the spine, which is incredibly small. And so what we're seeing is an increase in the postsynaptic density after fear conditioning and a decrease after safety conditioning. Safety conditioning is where the tone predicts the absence of shock. By doing this a bunch of times, the animal learns that no shock is going to occur. So with fear conditioning, you get an increase in the postsynaptic density. With safety conditioning, a decrease. So these are actual structural changes that we can see through these kinds of techniques. So the brain is changing physically during learning. When we learn, it's not just in our minds, it's in our brains, it's in our synapses, and here we are identifying specific synapses in the amygdala that show these structural changes. So one last area. Someone tell me the time. 10 minutes. So here we have the circuits we've been talking about. So the inputs are coming in from the thalamus and cortex, activating the reactions here, freezing, autonomic, hormones, and so forth, and the neuromodulatory networks, which can provide feedback here. So these we call emotional reactions because a stimulus occurs and a response is automatically expressed. But in an emotional situation, as I said earlier, we're not only reacting, but we're also motivated to take action. So this guy is watching the stock market crash in 2008, in October 2008. So he's going to run up to his office and change his stock portfolio before it gets too bad. He's going to take action here. So how do we study this in a rat? So we use avoidance conditioning. Now, avoidance conditioning was the primary way that fear was studied for many, many decades. And it didn't lead to very clear understanding of what was going on because it's a very complicated procedure. It involved two steps. One is the animal undergoes defense conditioning at the beginning, and then it has to learn to perform an instrumental response. So let's take a look at that. So the rat's in the chamber. It hears the tone. The tone is paired with the shock, so then it's going to freeze every time it hears the tone. But over time, it learns that if it crosses, it makes a movement, and then eventually it learns if it crosses, it can, when it hears the tone, it can avoid the shock. So it's learning two things. One, that the tone is associated with the shock, and two, if I move to the other side of the chamber when I make a response, that I won't get the tone and won't get the shock either. So obviously, in order to do that, you kind of have to begin to extinguish the freezing response a bit so that you can take a movement. But the fact that he's shocked will make him jump and move around, and he'll cross over just to avoid the shock itself. And when he does that, the tone goes off. So what the termination of the tone is a reinforcer. What it's doing is reinforcing this instrumental response that is performed. So the rat learns to perform this response to turn the tone off. Not to escape the shock, but to turn the tone off. And there are other experiments that show that this is the case, which I won't go into, so just take my word for it. So, as I said, you have to extinguish your freezing a bit before you can take action. You can't act if you're frozen in fear. So as the fear response is extinguished, the avoidance response is learned. Now, in contrast to what's required for the initial defense conditioning part, the lateral amygdala is required, but the central amygdala is not. Instead, the basal amygdala is required. So in what we've been talking about before, it's the lateral and central nucleus for the reaction learning, or for the association that leads to reactions. But for the use of that association to control actions, it's the lateral and the basal amygdala that's important. So CS-US get combined here, converge in the lateral amygdala. That association then goes to the central amygdala to control reactions and to the basal amygdala to control actions. So somehow this reaction circuit has to be inhibited in order to get the action out. This is too complicated. I'm going to skip it. But this is how we have begun to think about this. So here's the first part that we've already talked about. The basal, I'm sorry, I put the basal amygdala in a different position here now. The lateral to central is here now. Lateral to basal is here. The basal connects with the ventral striatum, the nucleus accumbens, and this allows us to get into these instrumental circuits in the striatum that control actions. Neuromodulators, norepinephrine, dopamine, acetylcholine, serotonin, are all released by the shock and by the CS after conditioning, so that it modulates all of these circuitry. And so we have these parallel systems for controlling reactions and actions, which we're able to take apart through these animal studies like this. And perhaps Liz will tell you about some of her studies that have done something very similar in the human brain. But I want to just make one more point here. In an avoidance study, usually there's about 20 to 30% of the animals that never learn, and these are what we call poor performers. So these animals simply couldn't learn to perform the avoidance response. So our hypothesis was that if we lesion the central amygdala, the animals would then be able to learn the avoidance response, because the idea was that these are the pathological rats. They're frozen in fear so bad that they can't learn to take action. So we lesioned the central amygdala, and to our surprise, even though they hadn't really been performing the avoidance response, they had learned how to avoid. And as soon as the central amygdala was lesioned and then they were tested, they started showing avoidance and then learned it even better. But they already knew the answer by the time we had made the lesion. So this is kind of an implicit form of learning where it's not even expressed behaviorally in a way. But it's very important because it suggests that if we can somehow rein in the central amygdala through drugs or through psychotherapy, it might allow more adaptive forms of behavior to proceed more effectively, and for therapy itself to proceed more effectively. So very important is that we can give these animals a simple drug like propranolol, which blocks the adrenergic norepinephrine system, and get the same result. The animals that can't avoid can learn to avoid if they have propranolol. Now, propranolol is often is classically used for high blood pressure, but it can also be used for stage fright and other purposes, and it can be used in some cases to treat aspects of post-traumatic stress disorder as well. So basically, we're kind of reducing the stage fright of the fear of the experimental context and the tone here in the rats, allowing them to then perform the avoidance response and to learn more effectively. So I just want to close with by saying we've done a lot of studies, which I don't have time to go into, but a lot of our work is emphasizing the importance of the studies of the animal brain for being able to improve therapy in various ways, both psychotherapy, behavioral therapy, cognitive behavioral therapy, as well as drug therapy. We do a lot of work on something called memory reconsolidation, which is the attempt to block the restorage of memory after it's retrieved, so that the memories then don't elicit emotional responses later. This has proved to be very effective in rats, both in blocking these fear kinds of memories, but also in affecting rats' craving for drugs and other kinds of things. So it's a potentially very important area of research. It's been shown by Liz and others to also work in the human brain. And so we do a lot of this work with an eye towards helping therapy proceed more effectively. And many other people in my field, such as Michael Davis, are working on this as well, trying to figure out how to make cognitive behavioral therapy more effective. So let me just close with a few summary points about some of these survival systems, or motion systems. They contribute to survival. They engage the whole organism. They synchronize and monopolize brain resources when active. They inhibit non-relevant brain function. So in a situation of danger, sexual behavior, sexual systems and feeding systems and other things are inhibited. But as I said, as motivational changes take place, in other words, as hunger increases to a certain point, then feeding has to then trump the defense system. But under most standard conditions, the defense system will trump almost everything else, because you have to stay alive or there won't be any more eating, drinking, sex, and so forth. So they put a conditioned stimulus potentiates these motivational systems, as Randy Gallistel said in his wonderful book in 1980, The Organization of Action. And then feelings are the conscious witnessing of our interpretation, witnessing and interpretation, and appraisal of all these organismic states and the environmental context and the memories and so forth that are being elicited. So these are some of the people that have contributed to all this, and thank you very much. And I apologize for talking so long, but they gave me a 2-hour talk, so it's their fault.
Thank you, Joe, for such a nicely tuned piece of scientific music.
Thank you.
And now we have some time for questions or comments, so please.
Jardine, you always have something to say. I'll pay you later.
So I encourage you. Well, please.
But that's not because of what you said. So it seems to me that the term appraisal is used in two different ways. One of them in your last slide, or the one before the last, you used appraisal as a conscious appraisal. But I think that appraisal is something more general that includes also implicit appraisal, which means the brain does it without you knowing. So can you comment on that, on the difference between?
Yeah, so I didn't, let's see how I used it in the last slide. So
Here.
yeah, so
Last one.
Right. So, but earlier I said these survival circuits are basically processing or appraisal networks that are tuned to certain kinds of pre-wired stimuli and generate these hardwired responses. So I think each of these survival systems is an appraisal system, but I think we should probably find a different word. So rather than appraise does sort of imply kind of it draws you towards the higher end of that concept. So maybe evaluation or something like that. But yeah, I agree that most of the appraisals taking place in the brain are unconscious, and it's only the conscious, the last steps that lead to feelings that involve the higher order cognitive appraisal.
Thank you.
Yes, please.
In some pathogenetic role, you mentioned some noradrenergic system and also glutamatergic system. And the most frequently used drugs for the anxiety disorders are serotonin selective serotonin reuptake inhibitors. This is serotonergic drug.
Right. I was just trying to compress things a bit, but we've done studies on SSRIs. One of the mysteries about antidepressant medications is why they take so long to act. Right? And so we tried to begin to address this question by looking at whether acute versus chronic treatment with SSRIs would affect this kind of defense conditioning procedure. And what we found was that an acute injection of an SSRI, citalopram, increased fear, or increased freezing in the paradigm, but a chronic injection over 20 days reduced freezing. And this is very similar, as you know, to what you see in patients where you can have increased agitation and anxiety at the beginning of treatment. So in addition, what we found was that the chronic treatment was associated with a change in the regulation of glutamate receptors over the long period. So this also is also consistent with what I think a lot of people believe, which is that the SSRIs need serotonin as an entry point, but their therapeutic effects have nothing to do with serotonin.
Thank you.
Please, yes.
Possibly.
Joe, regarding this last conclusion, feelings are the conscious witnessing and interpretation of the organismic state. Well, I know your research is not about that, but I do believe you may have some thoughts. if we look at at feelings from this perspective, that means that well, what we know about witnesses, that it changes what witness will tell you about the event.
Closer.
So, witness. If someone is a witness of an event, they may change their the report, the story. So, would you agree with the view that having the same or similar underlying organismic state may evoke different feelings in an individual across life?
Yes, well, the organismic state is, and the context, right? So the context can change, can interact with the organismic state to determine what it is you're going to feel, right? So you could have the same kind of, if we go back to the Schachter-Singer theory of emotion, where their idea was that in a situation that has it's ambiguous to the person, but there's physiological arousal that's been generated, the person looks to the environment to explain the organismic state that they're in. So, I think there's value to that point of view. Obviously, that's not the whole story about where feelings come from, but I think that that's certainly a component of it, that the social and physical environment in which an organismic state occurs will determine what you feel. So in your talk, you talked about if you're in a social context where a child and mother are together, that can reduce the fear response that the child feels or an anxiety response that the child feels when an intruder comes into the room, or a stranger comes into the room. So in that case, the mother is doing the social buffering. So the outside world in that case is modifying how the situation will be perceived and responded to.
Please do not feel dominated by the first row. But the first row, please don't afraid the rest of the room.
I'll just make one comment while we're waiting. But there are a lot, as we said at the beginning, there are a lot of emotion theories, and I think probably most theories are correct in about some aspect of emotion, and we just don't have a big theory yet that is going to explain everything. That's something I think that we still are waiting for. People like Nico Freida have done amazing jobs of trying to synthesize all of the psychological literature on this, but it's very complex, and we need to approach it from lots of angles, from the brain, from psychology, psychiatry, many angles. Literature, philosophy, all of them. And that's why we have this whole conference, which brings in the humanities as well as the sciences into the topic of emotion.
Some more questions and comments? Second row question.
It's not my line, but it's just my hobby. Question is, if cosmic rays, cosmic waves, affect functioning of brain and earth, and how quantum mathematics is applied to functioning of brain?
Michael?
Well,
I'm sorry, I can't even begin to answer that.
I think it's just like any other mutation.
Like a mutation?
Yeah, because it can change some
perturbation,
cause some perturbation in our brain.
Like cell phones?
And certainly cosmic wave played an important role in our evolution, because the Earth and all organisms were exposed to cosmic radiation from the very beginning.
Third row? All right.
Please.
No physics, please.
I just need on a form, to express my question in a good way. you were saying about based on the example of the phobia. What I understood that some parts of the amygdala can learn from that it gets from the outside world the stimulus, and that then it kind of changes the structure. And for instance, some very strong stimulus can modify it in a way that it stays, like the phobia. And I was thinking, if we know how it works and what kind of stimulus can modify it. For instance, we can also, in a way, influence the amygdala by positive emotions or any other things, and it will also modify the whole structure. So I was thinking, by knowing it, we also can assume that we can influence the brain, for instance, by language, creating certain chemical states, for instance, emotions and so on. And I was thinking, if we know that, how we can use this knowledge to kind of accelerate and to make it better and faster and more suitable the therapy, for instance, of disorders like phobia and that kind of emotional disorders. What would you say about that?
Yeah, so I mean, one of the things that therapy tries to do is to reduce fears and anxieties. And one way to do that is to expose the person to the stimulus that's harmful or that they're afraid of. So a patient who's afraid of snakes can be gradually exposed to snakes. First you go to the same building where there's a snake, then you're going to go into the room. The next day you go a little closer. Eventually you go very close. And through this kind of systematic desensitization, you then finally are able to, not be so terrified of snakes. So what you're doing is changing the way the brain is responding to the snake. So if it's a learned stimulus, you can do also counter conditioning, where you pair the stimulus with something positive rather than negative. You can try to change the memory of the stimulus, and that's something we're trying to do in our research, where, for example, people who are exposed to some sort of trauma, some small percentage, about 20%, 25, 30%, develop post-traumatic stress disorder. And so there are specific trigger cues that activate that, the symptoms of PTSD, some of the symptoms. And if you can reduce the ability of those cues to trigger the fear and emotional reactivity that they experience, then you can allow them to more successfully go through therapy because they're not so traumatized by these trigger cues. So we're trying to find ways to reduce the meaning of those stimuli and more effectively allow them to engage in other aspects of therapy. So I'm not sure I'm answering your question, but there's a lot of work being done to try and alter the way the brain responds to emotional stimuli in one way or another through kind of basic science studies, but then also try to apply that in various ways to studies of normal individuals and then to people with serious problems. And Liz and I have collaborated for many years on taking animal research to studies of healthy individuals, normal individuals, and then that because we have a solid understanding of how these things are working in the normal rat and in the normal person, we can then try to move this into the clinic and to some extent.
This is the last opportunity, since we have only 5 minutes left for our discussion. We have a very strong deadline this time.
James Russell.
Simple question. It's about the nature of extinction. If I understood early on, you talked about the connection between, and in phobias, the connection is retained in extinction so that it can be reinstated with phobia. Later, when you're talking about Hebbian mechanisms and these beautiful diagrams, you talked about weakening of the actual connection between the conditioned stimulus and at the postsynapse. So which of those is the nature of extinction, or both?
Well, so extinction is, when we were weakening, that wasn't through extinction. That was a different stimulus that is unpaired, so it signals the absence of shock. So that's not reversing anything. That's simply learning that the tone predicts safety rather than predicts shock. Extinction is, for the most part, thought of as new learning. So it's not the elimination of the old association, it's the creation of a new association that inhibits or overrides the previous one. And that involves connections between the prefrontal cortex and the amygdala.
So I think we should thank you once more.
Thank you. Can I make one more comment? I forgot to mention that there's been a lot of very good brain research in Poland on the amygdala. It was done in the 1960s. Elizabeth Fonberg, I think she was a student of Konorsky's. But if you're interested in this whole topic, she did many, many seminal studies of the amygdala, I think in dogs for the most part, in Warsaw in the 1960s and 70s. So.
And now we should join together to thank our audience. Thank you very much for coming here. And this is not the end of our encounter. The following is tomorrow. We start at 9 o'clock in the building of the Polish Academy of the Art of Sciences. So see you tomorrow at 9.